So I'm going to bring you back a bit to the bioinformatics field. There was some bioinformatics I saw in your presentation too with gene ontologies, but I'm going back to a bit more towards Pekka's talk. So the title is a bit changed compared to there was a mistake in the title in the program. But so this is work done in eNanoMapper. It was based on joint experience from the bioinformatics field between mainly two partners, Maastricht University and us at Misvik Biology and also Karolinska. And we tried to bring bioinformatics a bit closer to risk assessment. And so the basis was basically that toxicity data sources are increasing. As most of you know, there's big data, novel types of data coming out, and risk assessors need to adapt to this to make use of it. So data-driven modeling, applying bioinformatics is quite good for hypothesis generation and mechanistic insight, and to really make sense of pathobiological effects. But it's generally not applied in risk assessment quite yet. Sometimes for weight of evidence, but not quite that strongly yet. So, but the adverse outcome pathway concept has provided some means to apply this type of mechanistic data and biological data in risk assessment. But risk assessors still, I think, need tools for translating this AOP concept into reliable frameworks and strategies. So bioinformatics is based on a lot of data gathered along the last few decades, gene databases, protein databases, and also pathway databases. And this database interpretation of mechanisms is quite complicated, and to really make sense of it in the context of disease is even more complicated. And so databases sort of challenge that we noticed and sort of came up in the eNanoMapper project was that databases variably annotate specific genes to biological processes, meaning that different pathway databases can describe the same. So these are three different pathway databases, KEGG, Reactome, and WikiPathways, and they variably describe apoptosis so that only 10% of the genes in those databases are common to the same biological process. Now, this can be overcome bioinformatically if you really are experienced, but within risk assessment, it becomes impractical. And on the other hand, also within the same databases, there can be variable annotation of specific pathways to biological processes because of biological redundancy. There's different pathways describing the same type of process or even the same process. So here, WikiPathways is describing apoptosis again in 4 different ways, and only 4 genes, or 4% of the genes overlap between these pathways. So again, causing long lists of redundant pathways requiring further analysis or really expert interpretation to make sense of it. So the AOP concept sort of enables incorporation of this mechanistic data. And just to show what the AOP is, I don't know if everyone is familiar with it, but it's basically, instead of going from exposure to directly correlating it to outcome, an adverse outcome, we look at actual key events in the middle and try to use those as indicators of disease. So basically, it's a systematic ordering of key events, causally connected key events that indicate disease or adverse outcome. And so we use this to our advantage and generated this 6-step workflow where we integrate information, data, into a broader adverse outcome pathway linked molecular scheme, so a molecular pathway incorporating information from AOPs. And we used Wiki Pathways because it's a collaborative platform. It enables open use of pathways and publishing of pathways in the repository, and PathVisio, which is an editing tool for actually building pathways. And we gathered information on a disease, so looking at well-developed schemes of the disease in literature, looking for genes connected to the disease from databases. There's a lot of databases on this kind of information out there. Interactions between the genes and functional schemes of those genes, and then finally linking that information to AOP key events. And we used this workflow on a case study on pulmonary fibrosis, which is relevant to nanotoxicology, particularly due to multi-walled carbon nanotubes, but I think also other particles may be inducing this type of disease. And we looked in literature for general schemes. We found quite well, it's a well-studied disease, so it has a lot of information out there. Surprisingly, however, it doesn't have a pathway, a molecular pathway. There's no description of the disease on a molecular level. We also looked at genes. We found 64 very strongly linked genes to the disease. We looked at database access points, so information gathered from other databases linking the information together, so interactions between the genes and functional groupings of the genes. And then we also knew that there was a recently published putative AOP for the disease, meaning that it's not published as an OECD reviewed AOP quite yet, but it's underway, and it has described key events towards the final adverse outcome. And so this is what the pathway then finally looks like. It went through steps of modifications and really a lot of work to get it to connect nicely between the different elements. And what it does, it's bioinformatically employable, so you can use it for a bioinformatic analysis. And it connects basically the genes, so all these 64 genes that were related to the disease, and it also connects those genes to pathways which are actually interactively clickable, so you can go into other pathways that are reactive oxygen species metabolism or differentiation pathways, cytokine and chemokine signaling, and other types of pathways that are also known to be linked to the disease. And interestingly, after doing all of this, we could also then place key events. So they're only mentioned here now as key event 1, key event 2, and described up here. But eventually, when the AOP becomes a qualitative AOP or even a quantitative AOP, then these can be changed to computational links, which can be then clickable to go from an omics experiment to a pathway analysis and then directly into an AOP. So, and interestingly, recent thinking on toxicity testing actually argues for applying this type of pathway analysis using these AOP-linked molecular pathways. So Rusty Thomas was recently at the ECA and described this. They had a new approach methodologies workshop, and he envisioned this a type of high-throughput transcriptomic assay which enables screening of transcriptomic changes by many types of agents or nanomaterials, which would proceed to an AOP-based analysis, and that then continues on to a hazard characterization actually based on the AOPs, so directly giving information and hypothesis on the disease outcome. And we believe that the pathways could be useful for this analysis. So doing then different types of disease-linked bioinformatics analysis, instead of just looking at the mechanisms, you would look at the mechanisms, but also have the link to the actual final adverse outcome. And they're applicable, so the pathway would be, or pathways, eventually there would be several disease pathways, would be also applicable in QSAR approaches. We've been working with omics-based descriptors for QSAR, so generating gene sets basically useful for QSAR approaches. And also within the PTGS that Pekka just presented, it could be a means to describe the components that are predictive of certain endpoints. So leading to a final disease-linked grouping and read-across or possibilities. So to go back to a similar slide as the one that Pekka showed, this is, if you were here yesterday listening to Vesa on the high throughput screening, this work is also part of that sort of that workflow where we start off with high throughput screening. In this case, we then would think maybe that we could go into omics screening even on a higher throughput level, and leading that to bioinformatic analysis, which would be more disease linked, again, or AOP linked. And then finally, looking closer at the actual mechanistic validation of those findings in the omics data. So, in conclusion, do I have time for those? So basically what we studied was that a concept of data enrichment into AOP-linked molecular pathway descriptions, which could support weight of evidence-based and biological data-based nanomaterials risk assessment. And so we used this 6-step workflow to incorporate data, public data, which enriches AOP descriptions with molecular information, really showing, so we used this disease pulmonary fibrosis, and it resulted in a pathway that really describes the underlying molecular mechanisms and shows how the genes are interlinked, how the functions relate to the genes, which pathways, other types of pathways and databases are linked to that, and how the AOP events could be linked to that. So overall, we believe that it has a strong power of fusing data sources and applying bioinformatics-based tools, and really could enable novel ways of describing how nanomaterials cause cytopathological effects. So, and yeah, I'd like to thank the whole eNanoMapper consortium, actually, because even though it was based on bioinformatic experience or from 2 partners mainly, Maastricht University, Linda and Freddie were quite central in the actual building of the pathway, and us at Misvik and Karolinska, but also the QSAR modelers, Georgia and Harry, they've been central to driving this, because I felt like it really could enable some omics-based and really disease-linked omics-based QSAR modeling. And also Nina kind of gave me the idea to do the whole thing from the start, so yeah, thanks.